Good evening, Emma. Okay, so we were discussing the, you can say, various types of endosperm formation, nuclear type, the cellular type, the halobial type, and all. Good evening, Vanshika. So today we'll be discussing a very relevant topic known as the development of dicot embryo. Okay, like in Capsella bursa pastoris, known as the crucifer type. Good evening, Asma. So let's start with the dicot embryo development. So before going to start the dicot embryo development, I will try to tell you that there are multiple types, okay, of embryo development. So the crucifer type is what we'll be comprehending in the today's lecture. But what happens is the mode of origin of the four-celled proembryo and the contribution made by each of these cells makes the base for the classification of the embryonal type. Okay, so there are multiple types. Okay, so the terminal cell, meaning that if you have this cell in the terminus, okay, that is the, and this cell when it divides at towards the end, okay, into two, so this cell is known as, this cell is known as the terminal cell. Terminal cell. Okay. So this is the first transverse division in the zygote, right? This is the zygote. First transverse division in the zygote. So depending on the terminal cell, okay, of the two-celled proembryo, because I told you what is the proembryo. Proembryo means the two-celled zygote till the differentiation of organ stage in plant. These entire stages are known as proembryonic or proembryo stages. Okay. So number one, which will be or which is given in your course, is the crucifer type. Okay. In crucifer type, what happens is this is the basal cell. This is the terminal cell. So in crucifer type, number one, the basal cell plays little or no role in the development of the embryo. That is the crucifer type of embryo NCRT. So the basal cell and terminal cells both are there. So basal cell plays, you can say, little or no role, minuscule role in the development of the embryo. So that is the meaning of crucifer type. Okay, but crucifer is not the only type. Okay, we have multiple types that come in NEET examinations. I'll just acquaint you with that, but describe crucifer type later on in detail. But there are various types. We should at least know the name and the various characteristics of it. So the second type is known as the Asterat type. Asterat type. In the Asterat type, what happens is the basal and terminal cells both play, basal and terminal cells play an important role okay, in the development of the embryo. Of the embryo. That is point number one. Point number two, only for NEET examination, is the terminal cell of the two-celled proembryo. Okay, only talking about the terminal cell here. Okay, of the two-celled proembryo. It divides by a transverse wall. okay, divides via transverse wall. Means the transverse division takes place. Okay. Number three is the solenoid type. It is the SOLANAD, a very special type known as the solenoid type. So the solenoid type one, if you talk about, so what happens is the solenoid type, the basal cell in this type, okay, the basal cell in this solenoid type, it forms a suspensor cell of two or more cells, or it forms a suspensor, then there are also suspensor cells comprising of two or more cells. Okay. So when you talk about the suspensor cells, okay, so there are various divisions, okay, in plant which will lead to the formation of the suspensor cells. Always remember this type. So the thing is, if you talk about the suspensor cell only, okay, so suspensor cells are very very special cells. Okay, the plant embryogenesis, we know that it begins with an asymmetric cell division, always remember, and resulting in a small apical terminal cell and a larger basal cell. Okay, this is the fact that you should remember for your any kind of examination. So it is an asymmetric cell division always, because when I drew a diagram like this, okay, I wrote this is the terminal one, TC terminal cell, this is the basal cell. You can see which one is bigger. Okay. So what happens is it's an asymmetric division. It's not that two cells are equal in their volume or size. Okay. So the first asymmetric division provides polarity to the embryo. Okay, when we go deep, because this thing is not given in your real NCERT book, but it becomes really difficult without explaining this concept to go for any other concept. So this sets the, you can say, or provides the polarity to the embryo. So most of the plant embryo develops from the apical cell. Apical means the terminal cell. Okay, but the suspensor cells, they develop from the basal cell. Always remember, the suspensor cells like here, they develop from the basal cells. The suspensor, what it does is it anchors the embryo to the endosperm. Function of the suspensor, it anchors. Okay, this is also not given in many books. Okay, so note it down properly. It anchors, you can say, the embryo to the endosperm. The embryo to the endosperm and serves also as a conduit for nutrient for the developing embryo. A vessel of nutrient or conduit means transportation mechanism. It will help in that specific, you can say, type, means it will help the nutrient to travel. Okay, so it's a conduit for the nutrient. Okay, serves as a nutrient conduit, you can say, for the developing embryo. Developing embryo. Always know this function. It is asked in competitive examinations. This the function of suspensor cell you won't find in even reference books. Okay, if you purchase any reference book, ABC, Pradeep, or whatever you have, you won't find this specific. They will all draw suspensor cell. You will see, but the real meaning of suspensor cell, asymmetric division, those things will not be given. Good evening, sir. So the further cell division. Okay, now I'll be explaining. So now what happens is you have a further cell division. Okay, so now I'll talk about further cell divisions, but let me tell you about the other types. Okay, one more type I'll tell you is the carophylot type. Okay, carophylot type, type number 4. Okay, carophylot type. Just know these type names at least so that you can crack the comparative examination. Okay, so in the carophylla type, what happens again? This is a special type of division where the basal cell, okay, keeps on dividing. The basal cell keeps on dividing. After you have got an apical cell, that is the terminal cell and basal cell, the basal cell still keeps on dividing further. This is a very special characteristic of the carophylot type. Then last one is the chenopodiad type. Okay, chenopodiad type. Okay, so both basal and terminal cells take part in the development of the embryo again here. Okay, here also both the basal and terminal cells take part in the development of the embryo. Okay, so now we'll be going to the crucifer type again. Okay, where basal cell plays little or no role, okay, whatsoever in the development of the embryo. Okay, so we'll be taking the example of Capsella bursa pastoris. Capsella bursa pastoris crucifer type, which is the benchmark of your board examination. Okay, so for the first time, a scientist by the name Hanstein in 1870 he worked out the details of the development of embryo in Capsella bursa pastoris, which is a member of the crucifer. Okay, now what happens is here again the oospore or the zygote, it divides transversely forming two cells, a terminal cell, apical cell, or the basal cell. Okay, now the cell towards the micropylar end, this one. Okay, cell towards the micropylar end, top one, is the basal cell here, transverse division, apical cell or terminal cell, asymmetric division. Okay, this is a mitotic division again, not meiotic. Okay, mitotic division or equational division, mitosis. So the cell towards the micropylar end of the embryo sac, okay, is also known as the suspensor cell or basal cell. Okay, or it can divide to form many suspensor cells later on also. Okay, and the terminal cell here in this case, the crucifer type is responsible for giving rise to the embryo. Okay, so the terminal cell by subsequent division, okay, what it does is it gives rise to the embryo. Okay, while the basal cell it keeps on dividing, or it may not divide any further, it will keep on differentiating. Okay, it will keep on differentiating, you can say, and you can say synthesizes a proper suspensor cell or a proper suspensor. So that is what the aim of this crucifer type division is. Okay, so the terminal cell embryo, it divides by a vertical division. Okay, vertical division like this. This is now the vertical division. It will divide by, you can see, I'll show you the diagram if I have. That is much better than me drawing everything here. So that dicot embryo development. See many diagrams. You can see this only. This diagram is also good enough. Okay. So look at this diagram. So once you have a good look at this diagram, you will be able to understand what I am trying to say. Okay. The terminal cell, now watch this diagram properly. It divides by, you can say, a vertical division. Okay. Vertical division. You can see here vertical divisions are happening. Okay. Vertical division is happening. And vertical division leads to a four-celled stage, okay, or one-celled embryo. Embryo is one only, but you have a four-celled stage here, okay, 1, 2, 3, 4. So embryo is one, but four-celled stage, but one-celled embryo. In certain plants, the basal cell, it forms the hypocotyle. Now you will say, what is hypocotyle? Hypocotyle is nothing but the root end of the embryo. Okay, hypocotyle is nothing but the root end of the embryo in addition to the suspensor. Now the terminal cells of the four-celled proembryo, they divide vertically at right angle, okay, to the first vertical wall. They will form the four cells now. They will form the four cells. You can see they will be forming four cells here. Now after they have formed four cells, now each of the four cells, you can say, will now produce, each of the four cells will now produce, you can say, the, you can say, 8-celled stage via transverse division. Now you can say transverse division will begin like this. Transverse division will start beginning. Okay, transverse like this, horizontal. Earlier vertical was happening here. Now transverse division has started to happen. If you just think the switch from 4-cell to 8-cell stage means transverse division started happening. Very, very crucial here. Transverse division started happening. Now the four cells next to the suspensor, okay, the four cells next to the suspensor are termed as hypobasal or posterior octants. Okay, hypobasal or posterior octants. Okay, you can see here posterior octants. Okay, hypobasal or posterior octants. While the remaining four cells, they make the epibasal or anterior octants. Okay, you can say the next four will be called as the epibasal or anterior octants. Very, very important. Now, the epibasal octants, they give rise to the plumule and the cotyledons, whereas the hypobasal one, okay, whereas the hypobasal one, they will be able to, you can say, give rise to the hypocotyle, but with the exception of its tip. It will not give rise to the tip of the hypocotyle, but rest of the structure of the hypocotyle it will be able to give rise to. Now all the 8 cells of the octant will divide now, you can say, periclinically, which I had explained again, like transverse, not anticlinal division. Periclinically, which will form the outer and inner cells. which will form the outer and inner cells. Now the outer cells will divide further. Now outer cells will divide further by again anticlinal division. It will form a peripheral layer of epidermal cells known as the dermatogen. That is again a good concept. What is a dermatogen? So dermatogen, I'll write down. Dermatogen, it is nothing but the outer cells. They divide further by anticlinal division. Anticlinal division. Okay. Forming a peripheral layer of epidermal cells, that is known as the dermatogen. Now the inner cells again, they will divide by longitudinal and transverse division. Okay, the inner cells, they will again divide by longitudinal and transverse divisions. You can see here the dermatogen here. If you have your book, you can look at the diagram here also, because it is very complicated to draw on the whiteboard. Okay, so if you have the book with you, you can see it will be given in any good reference book, this diagram. So this is the dermatogen. Now you have something like the inner cells that divide by longitudinal and transverse divisions, forming the peribelum beneath the dermatogen and the plerome in the central region. Okay. Now peribelum will do what? The cells of peribelum give rise to the cortex, while that of plerome, it forms the stele. Very important. Okay. Very important thing here. I'll write it down. The inner cells, they divide further by anticlinal division. You can say, I should say longitudinal will be better here. Okay, longitudinal division by longitudinal and transverse division, because both go hand in hand. Only anticlinal would not be correct. So longitudinal and transverse division to form the peribelum beneath the dermatogen and plerome in the central region. Central region. Now, what happens is the cells of the peribelum, it gives rise to the cortex. Give rise to the cortex. These two sentences are very important. And while the cells of the plerome, the cells of the plerome, they give or they form the stele, give rise to stele. Okay, so always know the two things that which gives rise to cortex, which gives rise to the stele. Okay. Now at the time of the development of the octant, okay, whether you are going for the anterior octant, okay, posterior octant, doesn't matter. Okay, so you are going for hypobasal or epibasal, doesn't matter, posterior or anterior. Okay, epi means anterior, hypo means posterior. Okay, so at the time of the development of the octant stage of embryo, the two basal cells, okay, they divide again transversely, forming the 6 to 10 celled stage known as the filament. At the filament stage, okay, at the filament stage, if you see, at the filament stage during the octant they will form the filament, and at the filament stage what happens is the suspensor, it attains its maximum development. Okay, and it keeps on attaining maximum development until and unless, or you can say until the time the embryo attains the globular stage, which is given here, the embryo attains the globular stage. So by that time, the, you can say, the suspensor attains its maximum development, okay, forming the 6 to 10 celled filament stage. At that stage, concomitantly, the suspensor attains its maximum development. Always remember. Okay, so what happens is the further cell division leads to the globular stage. Okay, so the three basic tissue systems, you can say the dermal, the ground, and vascular. You might have read in class 11th. Okay, types of tissue. The dermal, the ground, and vascular. The dermal tissue, the ground tissue, and the vascular tissue. Okay, the three basic tissues, okay, tissue systems can be recognized at this point based on characteristic cell division patterns. Okay, then what happens is after this the globular shape of the embryo is then lost as the cotyledons, okay, which are the, you can say, the result of the embryo differentiation itself, it begins to form. So the formation of two cotyledons in the dicots, it gives the embryo a specialized heart-shaped appearance. Okay, the formation of cotyledons in dicots. What it does is it gives the embryo a heart-shaped appearance. A heart-shaped appearance. Very, very important. In monocots, what happens? Only a single cotyledon forms. Always remember. Only a single cotyledon will form. Now the distal cell of the suspensor is much larger than the other cells, and it acts as the haustorium. Okay, the distal cell of the suspensor, you can say this one. Okay, it is the largest cell, okay, or much larger than other cells, and acts as the haustorium. Haustorium. It acts as the haustorium. The suspensor pushes the embryo cells down into the endosperm. Okay, and the distal cell of the suspensor, okay, is much larger than the other cells, and acts as a haustorium. And the lowermost cell of the suspensor is known as the hypophysis. Okay, also know the lowermost cell of the suspensor is known as hypophysis. Okay. Known as the hypophysis. So you can see here, this is the lowermost cell of the suspensor. It is known as the hypophysis here. Okay. Now, by further division, the hypophysis, it will give rise to the embryonic root and root cap. Very, very important. It gives rise to embryonic root and root cap. So these are very prominent NEET questions. Okay. Now, with the continuous growth, what happens is the embryo becomes heart shaped, which I have already told you. Okay, which is made up of two primordial cotyledons. Okay, the mature embryo will consist of a short axis and two cotyledons thereafter. Now, each cotyledon appears on either side of the hypocotyle. Okay, so in most of the, you can say, dicotyledonous plants, the general course of embryogenesis is followed as what I have described to you in Capsella bursa pastoris. So this is the entire thing that goes on, okay, through the embryogenesis development or embryogenesis pattern. Okay, that is how we see or we analyze the different ways by which embryogenesis can occur or different stages of embryogenesis in the, you can say, cells known as the cells of the dicotyledonous plants. Okay, now if I talk about the monocot, okay, I'll show you the monocot also. Let's talk about the development of the monocot embryo. Monocot embryo. This site has a graphic diagram showing stages from embryogenesis. Development of monocot embryo. Look at Google images. Okay, look here. There are multiple cells. So this one is also included. I want that image to show it to you. Yeah, this image. Just look at this image. I will keep describing the image, okay, as we describe this entire process. Okay, monocot embryo. Now in monocot embryo, what the role is, the role happens or the switch reversal of the role. Okay, so what happens is there is no essential difference, you can say. There is no essential difference between the monocot and the dicotyledonous plants regarding the early divisions of the proembryo. Okay, but the mature embryo are quite different in both the groups. Okay, so the zygote, it transversely forms the terminal cell and the basal cell again. Okay, the basal cell, which is the larger one, okay, and lies towards the micropylar end, does not divide again. Okay, but becomes transformed directly into a vesicular cell. Okay, so I write it down. The basal cell, it does not divide at all. The basal cell, which is the larger cell and lies towards the micropylar end. Micropylar end. It does not divide. It does not divide again, but becomes transformed. Transformed to form, or becomes transformed directly to form a large vesicular cell. A large vesicular cell. Now the terminal cell, again it will divide transversely, forming the two cells. Of these, the lower cells or the lower cell would divide vertically, forming a pair of juxtaposed cells. Okay, forming a pair of juxtaposed cells, you can see here. Okay, then the middle cell divides transversely into two cell. Again, the middle cell will divide transversely into forming two cell again. Okay. In the next stage, the two cells once again divide vertically. Okay, to form the quadrants as we have already seen. Okay, the quadrant stage. Now the cell next to the quadrant also divides vertically, and the cells next to the upper vesicular divides several times transversely. Okay, that happens several times. It will divide transversely here, forming the octants. Okay, the quadrants now divide transversely to form the octants. The 8 cells, they are being arranged, or they become arranged in two tiers of 4 cells. You can say two tiers of four cells, they are arranged. Okay, now what happens is, with the result of the periclinial division, the dermatogen is completely formed. This is the dermatogen. Complete dermatogen is formed. Periclinial division happened, and one cell, then you can see the layering happens. I told you the layers will differentiate if you go for periclinial division. So the dermatogen is formed. Again later on, like the same process which I described to you in the case of dicotyledonous, the peribelum and plerome are also differentiated. Now all these regions formed from the octants develop into a single terminal cotyledon later on. So the peribelum, the plerome, all these regions, they develop into a single terminal cotyledon, because we are talking about the monocots here. Okay, the lowermost cell, okay, which is known as the cell L in some cases, lowermost cell of the three-celled suspensor. Okay, it divides vertically to form the plumule or stem. Okay, to form the plumule, you can see here. Okay, or stem tip. The cells which are known as the R cells, sometimes they form the radical. Okay, because you can see here some diagrams in there where you will be seeing the R cells, L cells also in some cases, like this one only. Some will be known as R, some will be this one where I showed you. See here, these kinds of things are there. The R cells are there, the L cell is there, the lowermost cell. Okay, so that's why I was referring to it as R and L. Some books will see R and L cell. Okay, so the thing is, the R cell, they form the radical. The upper 3 to 6 cells contribute to the formation of the suspensor again. Okay, so that is the specific thing behind the formation of the monocot embryo. Okay, so the R cells, they promote the formation of the radical, always remember, because in some cases you will see R and L system. Okay, RL, okay, mostly. So that is the L cell, on top you have the R cell. Okay, so that is the method by which the monocot embryo forms. Only the difference is the vesicular cell type gets formed, you can say, directly from the suspensor cell. Okay, so this is one of the differences, you can say, between the monocot embryo and, you can say, dicot embryo. So if you want to see the differences also, so I can write down the difference also, difference you can say between monocot and dicot embryo development. So if you see here, so if you look at the embryogenesis both, okay, so the main difference is what? The main difference, if we analyze, the main difference between monocot and dicot embryo is that the monocot contains a single cotyledon in the end, whereas dicot contains two cotyledons. Okay. One more thing is, if you see the basal cell in dicot embryo, okay, I'll just write down few points, you can write it down for dicot embryo. Okay. Dicot. So this difference will be of a lot of weightage for you, or importance also for you in the long run. Okay. Dicot embryo, if you talk about, so the basal cell forms a 6 to 10 celled suspensor here. Okay. In the dicot forms 6 to 10 celled suspensor. Point number one in dicot. Okay. Point number two would be the terminal cell produces embryo except the radical. Doesn't produce a radical. Terminal cell produces embryo except the radical. Point number three, the first division of the, you can say, terminal cell is generally longitudinal, that I told you. The first division of the terminal cell is generally longitudinal. Okay. Fourth one is it has got two cotyledons. So that I also have to write. Very important point. It has two cotyledons. And then the fifth one, the plumule is terminal. Okay. The fifth point is the plumule is terminal and lies in between the two elongated cotyledons. In between the two elongated cotyledons. So these are the 5 points which you should be remembering, or which you should be not remembering. If you have the concept, you can deduce from the dicot embryogenetic stage. Okay, the consequence of dicot embryogenesis. Now, if you come to the monocot embryo, The basal cell produces a single celled suspensor that I was telling you. It becomes directly transformed into the vesicular cell then. Okay, basal cell produces a single celled suspensor. Produces a single celled suspensor. Point number two, if you think of what it does, it forms the whole of the embryo. Okay, and also it is transverse in nature. Okay, there is single cotyledon. It forms the entire embryo. Okay, the terminal cell produces the embryo except the radical I was saying here, but the terminal cell here, it produces the entire embryo. The entire embryo. Okay. Now, the divisions are transverse, not longitudinal. For the terminal cell, there the first division was longitudinal if you see. So here the divisions are transverse in nature. Transverse division. That is one difference. Another one is, if you see, there is only a single cotyledon. A single cotyledon is only present, right? And the fifth point regarding the plumule, if we talk about, so the plumule appears lateral. Okay, the plumule, the plumule appears lateral due to excessive growth of the single cotyledon present. And there the plumule was present or is present between the two cotyledons. If you have noticed that I wrote down just few minutes ago, the plumule is terminal there and lies in between the two elongated cotyledons. Okay, and here the plumule is lateral. Just see the difference due to the excessive growth of the single cotyledon. So these are the main discerning points of difference. If you talk about the specificities regarding the dicot embryo and the monocot embryo, these are the only difference that you can think of. Okay, one more thing I'll tell you, in monocots like colocasia, no suspensor is formed. Okay, example is, some exceptions are there. In monocots like colocasia, no suspensor is formed. Okay, in Agapanthus, which belongs to the family Liliaceae, okay, two cotyledons have also been reported. Agapanthus, which belongs to the family Liliaceae, I can say Liliaceae. Okay, two cotyledons have been reported. Okay, so it's not a hard and fast rule. So you can see they have been two cotyledons reported also. Okay, now the terminal cell, okay, it undergoes number of divisions I told you in various planes and forms the single cotyledon. The single cotyledon is also known as the scutellum. Some cases the single cotyledon is also known as scutellum. Remember that also. Okay, now the middle cell I told you, it undergoes repeated transverse and vertical divisions. and thus differentiates into, you can say, few suspensor cells, the radicle, the plumule, and the hypocotyle. Okay, so in this type, the cotyledon is a terminal structure, and plumule is always a laterally situated, you can say, structure, or is, you can say, situated laterally in a specific depression. Okay, within the embryo sac when the embryogenesis occurs. Okay, so that is the, after the embryogenesis happens in the embryo sac, there is a depression produced laterally where the plumule will stay. So that is the concept behind the monocot embryo here. Okay, so we had discussed about the two types of polyembryony last time. Okay, the cleavage polyembryony and the embryos from the cells of embryo sac other than egg. Okay, so we have two more types of polyembryony. Okay, that I'll tell you for your NEET preparation. Okay, type number 3 is, if you check polyembryony, type number 3. So we have talked about the cleavage polyembryony. Okay, where we had the cleavage from the zygote or the proembryo. Okay, and we had the embryo from the cells of embryo sac other than the egg, means from the antipodal cells, synergids, synergid plus male gamete, all that, or you can say the haploid synergid cell becoming specific, you can say, zygote. But now the third type is embryos arising from cells outside embryo sac. Embryos arising from cells outside the embryo sac. Outside the embryo sac means what? You have the integument, you have the nucellus. So from those regions, right? Nucellus, the integuments. Okay. So what happens is the cells of the nucellus and integuments, you can say, have also been observed to develop into embryos. Both have been observed to develop into embryos. You have the citrus fruits, you have the mangifera indica, the mango. Okay, so they keep on happening in Spiranthes. The additional embryos have been reported to be developing from the inner cells of the inner layer of the integument. Okay, inner cell okay, of the inner layer of integument. Of the inner layer of integument. So that is the thing that I'm trying to convey. So now what happens is such embryos are very rare, but they are still produced. Okay, they subsequently come to lie in the embryo sac. Later on, they will traverse and get inside the embryo sac. Okay, and are nourished by the endosperm only. Okay, now embryos are directly also produced from endosperm type 4. Okay, type 4 are embryos are also produced from endosperm, directly the endosperm thing. This is also a probability and a specific mechanism, right? They have been reported in Belanophora. Okay. Now there were scientists who found that such embryos did develop from the egg, but they got embedded in the cellular endosperm. Okay, so that is the mechanism. They get embedded in the cellular endosperm later on. So it gives you a realization or observation that it is developing from the endosperm. Okay, then you have spontaneous and induced polyembryony. So induced one is completely artificial, spontaneous one is what I am showing you. But depending on that also there are two classifications. Okay, you have the gametophytic one under spontaneous polyembryony, you have the gametophytic one and the sporophytic one. Now, if you talk about the gametophytic one, gametophytic is all what we have already talked about. It arises from the gamete cell of the embryo sac. Gamete cell of the embryo sac. Two is the sporophytic. Now, if we talk about the sporophytic one, what I mean is, again, we have talked about this, okay, in the different types of true polyembryony. We have talked about this, like arising from the zygote, right? You can arise from the zygote. Okay, this is the generative fertilization of syngamy that they are talking about, right? Egg cell plus the male gamete. This is the gametophytic one. This is the polyembryony one, okay, the one that we have seen quite a lot, okay. So this one is the from the zygote, from the proembryo, okay, from the synergates, from the antipodal cells, okay, from the nucellus, from the integuments, okay. Nucellus integuments are the initial sporophytic cells of the ovule, you can call them, right? So that is known as sporophytic, okay. And I told you that it can also be done. This embryo development can also occur in the culture medium, okay. Then that will be known as induced polyembryony, okay. So in culture medium, if you develop embryos, they can be known as embryoids, supernumerary embryos, you can say somatic embryos, adventitious embryos, and so on. If you look at the plant tissue culture, which you may be studying if you become a botanist later on, but that is beyond the scope of this course, that to teach you this, you can say how does the plant tissue culture occur, the growth media and all. So mainly they what they do is they will mix up various concentrations of the plant hormones, auxin, gibberellin, and so on, cytokinin, and plus they will provide a special growth medium, special nutrients, carbohydrates, and all. So in order to promote the growth, okay. So I hope you are able to comprehend the lecture. In the next lecture, we'll be talking about the outbreeding devices, the pollen pistil interaction, okay, the use of the esterase, cutinase, alkaline, you can say alkaline, phosphatase, ribonuclease, okay, in the directly in the formation of the plant via genetic engineering, okay, pollen grain and tRNA rRNA ratio we'll be talking about. Then we'll be talking about the incompatibility, okay. So if you have any queries or doubts, you can definitely ask me on my personal number. I'll be replying as soon as possible. Okay, then thanks a lot.